is Sasha. She is a 10-month-old golden retriever. In our experiment, we will use Sasha to demonstrate the process of reception, integration, and reaction to a stimulus.
Now let's take a look at that again. But this time, we'll talk about what's happening in Sasha's eyes and brain, and the signal transduction pathway that links them.
When light enters the eye, it is first bent by the cornea. Then it continues through the liquid aqueous humor and through the pupil, after which it is bent again by the lens. It passes through the vitreous humor and hits the rods and cones located in the back of the eye.
Rods and cones are photoreceptor cells in vertebrates. Photoreceptors convert light energy into electrochemical impulses. Rods deal with light versus dark, whereas cones are responsible for the colors we see. But before we get into rods and cones, we have to talk about rhodopsin, a light-absorbing pigment. Rhodopsin is bound to a membrane called opsin, which changes shape when rhodopsin absorbs light.
The presence of light triggers a signal transduction pathway, whereas when it's dark, everything returns to normal. But if we threw a ball in the dark, Sasha wouldn't be able to see the ball, and then we wouldn't have much to talk about, now would we?
Right. So, the signal transduction pathway...
Yeah, so it all starts with the altered opsin, which activates a relay molecule called transducin. Transducin activates the second messenger, cyclic guanosine monophosphate, or cGMP for short, which is usually bound to the sodium ion channels in the cell plasma to keep the channels open. When the signal goes through, the rod cell membrane becomes depolarized and then releases the neurotransmitter glutamate.
And then, the cGMP is converted to GMP and the gated ion channels close, making the cell less permeable to sodium. This then hyperpolarizes the membrane potential. Hyperpolarization slows the rod cell's release of glutamate, which, depending on the signal, either excites or inhibits postsynaptic bipolar cells.
So that's rods. Cones, on the other hand, deal with color. Cones that form visual pigments are called rhodopsins. Those are your red cones, your green cones, and your blue cones. It's like the body's version of RGB colored TVs.
Don't they have yellow now too?
No, because there are three main wavelengths on the visible spectrum: red, green, and blue. All other colors are a combination of those three wavelengths, and therefore yellow is a mixture of red and green. Anyway, the red cones absorb red light best, the green cones absorb green light best, and the blue cones absorb blue light best. Why don't we move on to processing and integration of the information?
So, now we have to talk about the structures of the neurons. A neuron is a type of cell that contains a cell body, dendrites, and axons. Axons convey the outgoing messages to other cells. Neurons are coded in myelin sheaths for faster transport of signals. The cell that transmits the signal is the presynaptic cell, and the target cell, or receiving cell, is the postsynaptic cell.
Alright, now to integration. The information from the light signals goes from the rods and cones to the bipolar cells and then to the ganglion cells. The axons, or tails from the ganglion cells, come together to form the optic nerve, which heads straight to the brain. There are some other retinal neurons called horizontal cells and endocrine cells, which integrate the information before it gets to the brain.
The information is then sent as a series of action potentials along the optic nerve. There are two different pathways the information can take, vertical or lateral. In the vertical pathway, the action potentials go right from receptor cells to bipolar cells to the ganglion cells, which become the optic nerve. In the lateral pathway, the information goes from the horizontal cells to the photoreceptors and bipolar cells, which then give the information to amacrine cells and finally to the ganglion cells.
And horizontal cells inhibit distant receptor cells and bipolar cells, making light brighter and dark darker. This is called lateral inhibition. The lateral inhibition makes edges sharper and enhances contrast. This makes the image clearer and look more like what our brain sees.
As the information goes along the optic nerve, well, optic nerves, because each eye has its own, the nerves meet at the optic chiasm. From there, the information from the right visual field goes to the left side of the brain, and information from the left visual field goes to the right side of the brain, regardless of which eye it's from. The ganglion cells lead to the lateral geniculate nuclei of the thalamus, which continue to the primary visual cortex.
Information in the visual field is projected along the neurons by position, but it all arrives at the brain distorted.
So basically, the impulses from sensory organs go to the thalamus and from there to the cortex.
And the cortex, specifically the cerebral cortex, compiles and acts on the information that is given. The visual information is first processed in the occipital lobes in the back of the cerebral cortex. What we know as perception is the selecting, organizing, and interpreting of sensations by the cerebral cortex. After the information has been received and processed, the cerebral cortex directs the physical reactions to it.
While we're on the subject of responses, now might be a good time to talk about efferent and afferent neurons. Afferent neurons are the ones we've talked about already, the sensory or receptor neurons. They carry the impulses to the cortex.
And efferent neurons are motor neurons that carry the impulses away from the cerebral cortex, and they tell the muscles what to do.
Let's go back and summarize just a little bit. This is kind of a lot of information. The basic process is sensory input, integration, motor output. Sounds a lot simpler than what we've been talking about. We already know what the sensory input is. In this case, it's the visual stimulus of the ball being thrown. Integration is the process of interpreting the input and responding, which is carried out by the brain and the spinal cord. And then the motor output is the signals going from the central nervous system, or CNS, to effector cells, to muscles or gland cells that then react.
The process of sensory input, integration, and motor output can best be described as reception, transduction, amplification, transmission, and integration. Reception is, of course, the initial detection. Transduction is the creation of a receptor potential, which is a change in the membrane potential of a receptor cell. Amplification is the strengthening of stimulus energy that might be too weak to pick up normally. Transmission is the conduction of impulses to the central nervous system. And finally, integration, which is the processing of the information.
Well, that sounds a lot simpler than how we were talking about it before. Why didn't we do that first?
Well, I guess now it's time to see if Sasha ever did catch that ball. Roll tape, cue music, action.
How do I zoom out and in? Oh, hello. It was zoomed in really close to your face, and so it was all blurry.
It's okay.
Oh my goodness! Okay.
Let the gates close.
Yes, yes. Alright.
Flippers.
3, 2, 1. 3, 2, 1. Where's your cat? There are other retinal neurons called horizontal cells, and
Amacrine.
Trying to say that all over.
American cells.
America. It's America. Alright. Alright. Now to integration. The information. Stop.
I didn't do it.
You're holding your breath and I can hear. Alright.
What? No, we're not.
Stop.
I'm looking at the tree.
Alright. The verb.